he received his medical doctor degree from the same university. Now he served as a professor. And so after finishing his medical training, he went to US for his PhD degree. So in 2005, he finished his PhD study, and then he moved to the UPenn and also UCSF for his postdoc training. He back to Taiwan in 2008, and then he became the professor now at the Taipei Medical University. So now he is a professor, full professor, and also a director of that institute of the medical research, medical science. It should be say
clinical medicine.
Yeah, clinical medicine. So Dr. Tsai's expertise in the field of cancer biology, particularly in the subject of the multiple drug resistance and also metastasis. He has found several important gene and regulated pathways that can help the cancer cell away from the drug treatment. And one of the papers was published in the Nature Cancer last year. So that is a very prestigious journal with impact factor more than 20. So it's very important finding. And also that he has found several interesting proteins that can help the cancer cell to become metastasis. And so I think that the cancer cell is quite smart. He knows how to model a very diverse way to escape the immune system, also the drug treatment, to avoid the cell being destroyed by those external stress or external drug treatment. So there's a lot of new things which need to be explored, and Dr. Tsai is an expert in this field. And so I believe that he will give us very interesting talk today, and also everyone will enjoy his talk very much. So let's give a big hand to Dr. Tsai.
Thank you. So thank you very much, Director Chen, and I appreciate the opportunity to speak here at this prestigious institute, and also I have a brief chat with the faculty here, and also I see there are many, our students, I guess, and I think, it's a great opportunity for interaction and maybe more brainstorming of how to tackle the problem of cancer progression and late stage cancer, which is actually the real killer of cancer patients. And so as Director Chen mentioned, I actually, my career, I was a resident at the hospital next door 25 years ago, and I actually had multiple interests in medicine, not only cancer. I focused on cancer not until I did my grad study at Harvard because of the grant, because if you work with cancer, it's easier to get grants. But before, I'm more interested in information because I was trained as a GI specialist, and 25 years ago, I focused at the disease called pancreatitis. And so I collaborate with a professor, you're a retired professor, Dr. Wei Yaohui here, to do some analysis in mitochondria. So that's how I started my career. So I was back to the origin of my research. So I'm quite honored to be here. And so instead of talking about the other protein I have been working on for more than 10 years, which was luckily published in Nature Cancer last year, I spoke to Director Chen. I work with another, maybe even more interesting, more important protein for more than 10 years. And there are many questions remain, although we have cracked some of them. And I really look forward to the opportunity of interacting with the biochemists here to be able to go on and to dig more, maybe therapeutic or diagnostic opportunity based on the finding we accumulated so far. So this is actually a protein. Do I have a pointer here? So this is actually, so this protein is huge. 34, more than 3,000 amino acids, 409 kilodalton. So there was a joke when I graduated, and there was a postdoc, Japanese postdoc, he warned me. So Kelvin, my English name is Kelvin. If you want to start a career, don't work with something bigger than 250 kilodalton. So I was so lucky to happen to work with one protein called NCO2, which is 270 kilodalton, a bit bigger than 250. And this one is even bigger, 410. So I don't know. It's sometimes it's a fate. It's sometimes to do with a fate. When you came across something very interesting in your career, there is something is enticing you to go on. Although it might be difficult, but sometimes you will have some unexpected finding if you keep working on that difficult subject. So that's something I can share with the student here. I mean, don't always take the advice from other people. Sometimes you have to build your own interest and your own vision to go on. And this protein, it actually, because it's so big, it's very hard. Nobody knows about the structure of this protein. And so how can I get the structure here? This is based on the AI tool called AlphaFold. I think you guys should be familiar with that. So this is the predicted structure of this protein. And this protein is called ASP. I will introduce to you guys later. And this protein turns out to be very important for all aspects of cancer progression, including how cancer is going to grow and invade the surrounding structure or become metastatic to distant organs. So this cartoon just give you an idea of how many different pathways are controlled by this protein and which eventually lead to cancer progression and metastasis. So anybody know about the name Medusa? Have anybody watched a movie called Crash of Titans? It was a movie played like maybe 10 years ago. Not a very, not a hit movie, but interesting. It's based on the Greek mythology. So this witch, maybe, her name is Medusa. So everybody have been seen by the witch will turn to a stone right away. Anybody interested in the Greek mythology or history? And on her head there are many snakes, everywhere on her head. And so I mean, if we, as an oncologist, I think the late stage or like non-operable cancer is something like this. So it's a very complicated phenomenon controlled by many different aberrated pathways. And the take-home message is that we can sequence the cancer that we using the most state-of-the-art tools, maybe high-throughput sequencing, single-cell sequencing, whatever you can use. But what you get is a very complicated network of aberrated pathways. And if you say, maybe, I say there's one pathway called Win, which is important for cancer progression. And maybe the researcher or the drug company will try to develop an inhibitor for this pathway. But if we think the pathway stands for one snake here, if you kill a single snake, you still have many others to kill, right? And this witch still can turn people into stone, right? So, and maybe the other pathway might be important in certain contexts. And so the drug company, the researcher sometimes have luck in freeze this witch for a while. That's something we call targeted therapy, right? But eventually the other pathway will take over and become, the master regulator at the later stage. So that's why cancer always comes back, because there are so many pathways they can take advantage of. So, I mean, if we really want to cure or control cancer, obviously, I mean, focusing on one pathway might not be the ultimate answer. That's from my perspective. So do we have any even better way to control cancer? And meaning that, do we have any way to control all the pathway altogether? That's my question. To be able to do that, we need to know if there's any node or hub that can control all the different pathways in cancers, right? And anybody have any thought? Do we know of any hub or node which are already being used in clinic? Yes, indeed. The one of the most famous hub is PD-1, PD-L1, control all the different immune pathways in normal cells and also in cancer cells. So if we know about the hub, like PD-1 or PD-L1, and if we invent an antibody or maybe small molecule to dismantle or to disable that hub, we suddenly can move the field forward, right? That's what happened in the past maybe 10 years. There are so many different PD-1, PD-L1 or other checkpoint inhibitors that already been put into clinical use. But obviously there are other hubs over there in cancers. Maybe there are hubs in control cancer stemness or cancer stem cells, like in this case, and there might be other hubs that control tumor microenvironment, like fibroblasts or how the tumor infiltrating macrophages. There are other hubs. You can call them checkpoint. Right, so this is the very interesting one of the hub I imagine might be important for how we can move the field forward. So, and the story actually started from when I moved back to, initially when I moved back to Taiwan, I joined NHI, 国卫院, in 2008. And I was given a task because when I did my postdoc at the UCSF, we developed a model, something called 3D culture. And now the field has moved into the field called organoid. Quite, fascinating field. But back then, 15 years ago, they are not a very, trendy, concept. So the concept is that if we want to study cancer or tissue, you cannot study the cell. You cannot just simply take the cell from the context and study the cell alone. You need to put the cell into the context, meaning that you need to have the structure or organization similar to the in vivo scenario. So if we want to study cancer or tissue or development, we need to start from something like similar to the real tissue or cancers, meaning that they have to be in 3D, not 2D, right? So this is what 2D culture or 3D culture is used for. So we were able to figure out a culture scenario to force the normal epithelial cells to differentiate into normal-like structures, like in this case, breast, acines, or gland. And if we have some oncogene or tumor suppressor gene mutations, you will lose the ability to form structures, and they form actually disorganized structure called organoid or tumor spheroid. And you can apply the principle to different cancers, especially when the cancers are coming from glands. What cancers are coming from gland? Prostate cancer, breast cancer, and pancreatic cancer, many, maybe more than 50%. Even lung cancer is actually from, if you think of the alveoli, it's actually something similar to a gland, right? Colorectal cancer, actually the crypt structure in colon or small intestine actually are a secretory gland. So the majority of solid tumor are actually granular cancers. So we were able to recapitulate the process, and you can do anything you want. If you can recapitulate the process, you can do the profiling, like gene profiling or proteomic profiling of the cells before they form structure or after they form structure, and you get a lot of differentially expressed genes. And based on these differentially expressed genes, you can try to trace back to see what particular upstream signal are controlling the dysregulations in normal tissue or cancers. And the purpose is really to identify maybe novel targets or novel pathways that control how the mutated cell become cancer. So this is one paper we published in gastroenterology, and this is the most high-ranked journal in my field, in the GI field. And we actually put the pancreatic normal pancreatic stem cells in the 3D matrix and allow them to form pancreatic ducts. And so these are the ducts we saw from that model. We can actually recapitulate the branching ducts of pancreas. And you can actually, if you took the specimen from the lumen here, it's a tortuous structure. You might be able to see it's not a straight tubule or duct. It's actually tortuous. So you won't be able to see the duct, but there are real ducts in these structures, and there are something that are secreted into the ducts, just like what the normal pancreas do in our body. So we did the array, expression array, and we identified something called signature, which stands for a combination of genes and whose expression pattern can be correlated with the clinical, progress of the cancer, of the disease. It's called signature. And so we got like 12 gene signature, which can beautifully predict how the prognosis of the patient with pancreatic cancer. So, for example, in this cohort, if the patient have carried the expression pattern of this signature, the survival is very short, as opposed to the patient whose cancer does not carry this signature. And of this signature, we look at this signature and to see what particular gene we can focus on. And the first one is not very interesting because it's a lipid transport protein, and I myself is not particularly interested in like transport. So I turn to the second one, which is our theme today. The second one is called ASPM, and which stands for ASP. It's a Drosophila protein homolog in humans, microcephaly associated. And if you look at the gene ontology, it's categorized as a mitosis-related gene. So initially, it looked quite not very exciting or interesting, but this is the only single gene which interests me at that point, because if you look at the third gene, they are again lipid metabolism, ion transport, which is not my expertise and not very interesting to me. So I have no other choice but focusing on this protein. And so what is ASPM? So when we look up the literature about ASPM 15 years ago, it's not a protein study in cancer field. It's a protein study in neurology field. This protein is one of the many called MCPH genes that control how big your brain is. And it's a number 5 gene identified along the way. So the other name of ASPM is MCPH5, and it was initially identified as a centrosome protein, control the neural progenitor cell when they when they divide, they control the mitotic spindle process. So that's what we know 15 years ago. And I was impressed by the size initially. As a Jew, am I going to really work on this gene at all? So, but anyway, I mean, I will tell you later why I still focus on this gene now. And this is one of all the MCPS gene. This is the most commonly cause of hereditary or autosomal recessive mutated gene of human microcephaly. And this gene is actually been studied in the neurology field by a couple of famous groups, and people figure out how it can control mitotic spindle, because it actually control some particular protein that it's important for microtubule disassemble during mitosis of neural progenitor cells, and it's also important for the cell cycle progression in neural progenitor cells by controlling cyclin E protein levels. So this is what the patient with microcephaly look like. And So if you have kid, you have to bring to your pediatrician to measure the brain size, the size, right? So why? Because it's a rare mutation in one of these MCPH genes. The brain will become smaller than the normal kid. The normal kid, and this is what the CT scan of the patient with microcephaly look like. The structure are intact, but the thickness of the brain is much smaller than the normal brain. Here, these are the patient. They survive well. They can live, and their IQ is much lower than normal person. So this is what we know about this gene in the field of neurology. And the things get interesting later. It turns out there are a couple of groups identify that the evolution of this gene, ASPN, he undergo very rapid evolution during the past 350,000 years. And some region, they undergo very rapid evolution from the great ape, from monkey to human. And it looks like this gene, the evolution of this gene, has something to do with modern human, especially the modern civilization. So there's a paper, a very controversial paper, and published in 2005 in Science, and by a professor at Chicago, and he tried to correlate a certain mutation called haplogroup D of ASPN with human wisdom. And he said, he identified there are some region, particular region, their ASPN gene has a particular mutation, which is majorly localized in Jews in Israel and some region in Eastern Europe. But the region with the lowest incidence of this haplotype is in Africa and in some Southeast Asia region. And it became a very highly controversial paper because it seems to discriminate human wisdom, human IQ. But later on, people started to try to correlate the IQ test with this haplotype, and it turns out to be not correlated. So this is still a very highly controversial field. So after reading so many papers, it really got my attention. This gene is really interesting in the field of evolution and neurology development. So maybe this gene might be important for cancer development as well. So that's why keep me, focusing on this gene. And this is what we know. People know about some of the function of this gene, and the product of this gene, they can control cell cycle in neuroprogenitor cells. So this was a paper published in Nature Communication. They figure out in neuroprogenitor cell in Drosophila, the gene they interact with cyclin E complex and protect cyclin E from interaction with its specific E3 ligase, SCIP2 and FDW7, and thereby they prevent the cyclin E from proteasome degradation and stabilize. So if you have this ASPN there, the cell cycle can progress. But in the patient or in ASPN knockout condition, the cyclin E level will become destabilized, and the cell cycle will be postponed. The something called R point, meaning the point that dictate G1 to S progression will be delayed. And that's one of the reasons that explain why the mutation or deletion of this gene will cause the delayed division of neuroprogenitor cells. And this is another study published in Nature Cell Biology in 2017. And this is another function of this gene. This gene ASPN interact with an important protein called catenin, and during mitosis in neuroprogenitor cells, and this gene ASPN they interact with catenin and to form a complex, And what this complex do is during the later stage of mitosis, the complex sever or cut the mitosis spindle, enable to the cell to divide during the later phase of mitosis. So without this gene or with the mutated gene, the process cannot go on normally. So this is another function of this gene. I'm going to tell you tons of other functions later. So when I go back to pancreatic cancer, so I say, yes, this is a huge protein, but maybe it's important. Let's do something about it. And so I pick up this gene and do some clinical analysis. It turns out this gene the expression level is already very prognostic, meaning that the patient with a tumor, pancreatic cancer with tumor is high expression of this ASPM, the survival is much shorter than the tumor with a low expression level of ASPM. And this again, I think we did the IgC study, and the same thing with the RNA. It's highly prognostic. And then, is it limited to pancreatic cancer? If you look up, when you look up the TCGA, ASPM is upregulated in almost all cancers, solid tumors. So these are the comparison between pair normal, the tumor adjacent tissue and tumor tissue. We saw this expression is majority of cancer, they upregulate this protein. So it must mean something, right? Why cancer like this protein? So we go on to do some very preliminary cell studies. We knock down. So we first compare the protein. Actually, ASPM is up. Indeed, it's up in cancer cell lines. And if we knock down the ASPM using RNAi, and we saw that we can reduce cell proliferation and how fast the cell migratory assay, and we put the cell into mice, and we do something called orthotopic. So if you do pancreatic, you cannot do the subcutaneous model because it doesn't reflect the real scenario. You have to implant the cell into pancreas. So this model is called orthotopic model. So if you knock down the gene, the cancer progress slower than the wild type tumor, and less complications, shorter survival as expected. And so, if you remember, the ASPM is only one of these MCPH genes that control brain size. It turns out we are not alone. There are other MCPH genes that also have something to do with cancer. So there are actually 25 of them there, and ASPM is the number 5. So there was a review actually coming out last year, 2 years ago, and other people reported the oncogenic role of the other MCPH genes initially identified as the controller of brain size. So actually, ASPM is not the only one in the process. So we get excited. We are not we are not proposing something completely new. Nobody have never mentioned before. So I'm going to go really quickly later because today is about collaboration and exciting progress of the project. So, and all the paper except the last part is not published. All these papers are already published, and if you're interested, you can look up the literature to see if anything you might be interested in. So the first part will be our knowledge about the role ASPM in Wnt signaling. So go back to that story. So we want to figure out why ASPM is important for pancreatic cancer progression and prognosis. So we did the array, and we did the pathway analysis. And the number one pathway pop-up is Wnt pathway. So if you knock down ASPM, all the Wnt reporter in different cancer cells, they kill the reporter activity at all. So we were asked by the reviewer because cancer immunology has an impact factor of more than 30. So they want to know what's going on in detail. So we have no other choice. We try different way, RNA level, protein, co-IP, everything we can do within half a year. And we identify the real mechanism why ASPM is controlled Wnt. And there is an upstream regulator called Deshevil, which control the Wnt pathway. And what ASPM do is that it interact with DVL, in this case DVL2. There are 3 DVL 1, 2, 3. It interact with DVL2 in pancreatic cancer cells. And similar to what people have reported in cyclin E, it interacts with DVO2 and prevent it from binding to its own E3 ligase, thereby preventing the proteasome degradation. So if you knock down ASPM, you saw a lot of protein polyubiquitination. And the other downside of working with this protein is that we don't have any tool to overexpress this protein because the protein is so huge. There is no way to overexpress it. So everything we have to do the knockdown or knockout study. So that's the disadvantage of working with a protein of this size. And we know when it's important for stemness or stem cell. So in cancer, there's a theory that some aspect of cancer is controlled by stem-like cancer cell or something called cancer stem cell. So in pancreatic cancers, we saw that if you knock down ASPN, the cancer stem cell population, they drop. And this is an assay called tumor spear assay reflect the cancer stemness or the ability of cancer stem cell. If they exist in cancer, they are able to form 3D spheroid or organoid without attachment to the plate. So this is the very typical assay for analyzing cancer stemness. So if you knock down, you pretty much the cancer cannot form the so-called organoid. And then we turn to other cancers. So if this is true, it won't be only limited to pancreatic cancer. So the second paper is published in Oncogene. I'm sorry. Same thing. So in prostate cancer, the ASPN expression goes up, and in bone metastatic cancer, prostate the expression will go further up, and the same observation at the protein labels. And in this case, I'm going to skip really quickly to almost the same thing. So we come up with a model. The only difference is that I don't have I don't have the model here, but I have the model maybe later. So the only difference is that the other, remember there are 3 DVL. In this case, in prostate cancer, it's DVL3 are involved. It's regulated by ASPM. Very interesting. And the other case is liver cancer. And in this case, is the other DVL, called DVL1, is controlled by ASPM. So ASPM, they interact with DVL1, preventing its binding to the E3 ligase complex called QLN3 and KLHL12, and therefore stabilizing beta DVL, and in turn they stabilize beta-catenin, and to promote the target gene expression and eventually promoting liver cancer progression. So this was published in Stem Cell Report, and so this is the model I'm talking about. So we come up with an interesting model. So we think that ASPN, when they interact with DVL, it can protect the DVL from interacting with something called destruction complex. And what the destruction complex do is it degrades beta-catenin. But in the presence of ASPN, it interacts with the DVL, thereby they can they can stabilize beta-catenin, which can then translocate into nucleus to turn on the target gene. So without ASPM, the DVL will be degraded, so there won't be DVL available to pull the destruction complex to the membrane. Thereby, the beta-catenin will be degraded, and the cancer, won't progress because the target gene won't be turned on. So this is the first model I'm going to present today. And the second one is that things get more interesting. There are 2 major isoforms of this protein ASPM. And there are 4 predicted isoforms, but only the isoform 1 and 2 are expressed in cancers. And the only difference of isoform 1 and 2 are in the one particular exon, very long exon, exon 18. And the isoform 2 is a splicing variant of isoform 1, and it skipped exon 18 and becoming a much shorter protein here. And this is a review article we just, I think we're going to publish really quickly in Cancer Research later this year. And this is another paper. What we know about the idea about isoform is that from this paper in Journal of Pathology, we try to develop our own antibody to probe the ASPM protein. And we always come back with two different bands. One is the predicted band is 408 kilodalton, and there's another much shorter band, 250 kilodalton, and meaning that there are two potential isoforms using our antibody. So we did a lot of study to try to characterize their distribution. Turns out only isoform 1 was upregulated in cancer. I think it's here, right here. If you look at the pancreatic cancer cells, this is normal pancreas cells, no expression of isoform 1, but a lot of expression in the pancreatic cancer cells. But isoform 2, they stay the same. Very interesting. So we did more study, and we developed we developed our own antibody to recognize different isoforms. So we were able to see, the different expression level and different, distribution of these different isoforms. And isoform 1, for some reason, they tend to localize to cytoplasm. And isoform 2 is mainly in the nucleus and the perinuclear region, using our own antibody. Nobody was able to do this because they don't have the antibody available. And then we saw only the expression level of isoform 1 is prognostic in pancreatic cancer. And the expression of isoform 2 had nothing to do with how long the patient is going to live or survive. So this is another paper we published in Gastric Cancer 2 years ago. And this is another, so we are trying to identify the upstream regulator to regulate ASP and the isoform 1. And it turns out there's another stem cell related protein called FOXM1, control the transcription of ASPM. And this gene, this protein is very interesting. It also has other function. It can interact with beta-catenin and to pull beta-catenin into nucleus. So we are showing actually a loop here. This upstream regulator ASPM, they on one hand, it promotes the expression of ASPM, and which in turn promote the stability of DVL and beta-catenin. And on the other hand, FOXM1 interact with beta-catenin to enable beta-catenin to translocate into nucleus to turn on Wnt target gene. So this is making the pathway even more complete in this paper. And the second part, I think the third part is our recent finding is that we're thinking that if ASPM is such a huge protein, it tends to stabilize its binding partner, right? It stabilizes cyclin E, it stabilizes DVL, and there was another paper which caught our mind is it also stabilized something interesting in cancer, BRCA1, DNA damage repair. Exactly the same thing. So we are thinking about perhaps if it tends to do something to other binding partner, maybe it will do something similar to other developmental pathways. There are many important developmental pathways other than Wnt. And there's a pathway called hedgehog, right? Anybody heard about hedgehog before? And I think this one is even more interesting, Notch, right? Everybody know about Notch. There are 5 to 6 important development pathways important for cancer progression. So we screen all of them to see what ASPM might do to all these different development pathways. Turns out we got luck. Bingo. So for this paper was just published in Cancer Research last month. And what we figured out is that there is a important pathway in small cell lung cancer, hedgehog pathway, important for small cell lung cancer progression. Small cell lung cancer is pretty unique. It's one of the neuroendocrine tumor. It's not from, the origin of the cancer is not from epithelial cell. It's from neuroganglion cells. It's a bit different from pancreatic cancer or prostate cancer or liver cancer. But still, ASPM is important for the progression of this cancer type. And what we show is that if you knock down ASPM, again, the cancer won't grow at all. They won't have cancer stem cells. And then we did the reporter array to screen what other development related pathways are also reduced when we knock down ASPM. If you look at the ranked list, number one, if you look at Wnt is not actually very high in the ranking. Number 1 is hedgehog in small cell lung cancer. And if you go down, their number 3 is notch. Win is actually ranked number 5, not very high. So we were very excited. Maybe hedgehog is even more important for the role of ASP in cancer than the Win pathways. This is the case. So it took us, so Professor Xu, who used to be my postdoc, worked very hard on this project. So and also I have a postdoc, Lacey, who wasn't here, worked very hard on this project. And we find out that what happened is that, I have a model here. So ASPN isoform-1, they interact with the transcription factor of hedgehog signaling called GRI1, and does exactly the same thing to GRI1 as what it did to Destravel or cyclin E. It interact with GRI1 and prevent the GRI1 interaction with its E3 ligase, Culin-3, and beta-TRCP, and thereby stabilizing the GRI1, and therefore the signal can transduce and to turn on target gene expression. And ASPN also did something interesting in the hedgehog pathway. It also can stabilize an important membrane regulator in hedgehog pathway, something called smoothen. This is one of the most famous stem cell factor we know we use to sort stem cells. So it stabilizes the smoothen expression at the RNA level indirectly through the beta-catenin pathway. So we are seeing the intersection of 2 different pathways. One is Win, and the other is hedgehog. And ASPN seems to be promoting both, right? And so now we have another paper under review now in oncogene. This is about Notch. So I don't think we have time for that, but let me show you how it did something important for the important, here. So in this case, in liver cancer, other than Win, it also does something to the Notch signaling. So ASPN isoform interact with the intracellular domain of Notch1 called NICD1, and preventing it from binding to the E3 ligase called FBXW7. So thereby stabilizing the receptor, and which allowing to turn on the Notch target genes to promote liver cancer progression. So very interesting, right? It seems to do the same thing to different binding partners and different pathways, but it turns out these pathways are all important for cancer progression. So it reflects my second slide. It may fulfill the definition of the cancer, the hub or not. If we have something, if we can develop some way to, inhibit this protein, we might be able to, inhibit all the pathway all together. That's my dream. Right. So I mean, there's more data. I'm going to skip this. So I'm going to skip really quick. So other group, there's a group in China. They identify that ASPN is promoting DNA damage repair by stabilizing important, double-strand break repair protein called BRCA. Right. This is a famous gene in the cancer field. And so this is the model presented in our review. So ASPN is a huge protein. For some reason, it interacts with different binding partners, preventing them from E3 ligase mediated degradation. And some of them are isoform dependent, but some are dependent on other domains. So this is still a mystery to me. I mean, why it does the same thing to different partners through different domains, or maybe there's no domains. We don't know yet. And this is the last part of my talk, which is unpublished. And we actually spent almost more than 8 years, right, Mark, on this project. So other than this development or DNA damage repair or cell cycle proteins, they all are important for how cancer can progress and grow, right? But actually, 90% of the cancer patients are killed by cancer metastasis instead of the primary tumor. And it turns out ASPN is also play an important role in cancer metastasis, especially the metastasis related to cancer stem cells. So this project is a completely new, we identify a completely new protein complex, and there are certain important normal cell process, something called polarity control, which was initially identified in Drosophila. And the normal epithelial cell, they use the polarity complex to control where their upside, where their bottom are, where are the lateral region of the cell, right? They are a group of polarity protein complex that are controlling the mechanism, how the cell know its direction, right? For a gland, for like, if you are breast epithelial cell, you need to know where you are going to secrete your milk, right? If you are pancreas epithelial, you need to know where is my top. I need to secrete maybe amylase or lipase, right? We need to know where the lumen is, right? And where is my ground, right? The basal layer, right? And where is my side, right? So there is a group of proteins which is controlling the process. And it turns out ASPN is important to control stability of some of the protein complexes in this process. And these processes are actually important. So here is the breast cancer. If you knock down ASPN, pretty much you kill all the cancer metastases. But this is very ideal, right? You knock down the protein in vitro and put the cell in tail vein injection, right? And so we actually are able to see that the ASPN isoform-1 are particularly upregulated in cancer stem cells, and in the particular subset of cancer stem cell that it's important for cancer metastasis, in this case, breast cancers. And this is one of the complex we identified. It's called PCP complex. And I hide some of the identity of protein, but this is unpublished data. We spent 8 years on this project. Completely new complex. But it's very difficult to figure out what's going on in that complex. We're still ongoing, but we are almost there. And it controls the complex. The role of this complex is control a structure called invadopodia, or the other nickname is called cellular feet. What does it do to normal cell? In normal cell, like immune cell macrophages, when they move, they need to secrete something to digest the extracellular matrix, right? And to push the matrix to allow the cell to invade or migrate to different places. So this is a basic cellular machinery. But cancer cells, especially cancer stem cells, they tend to take advantage of this structure to allow them to invade and metastasize to distant organs. This is one of the key mechanisms for cancer metastasis. And ASPN is controlling this process. So, I mean, again, it actually supports the role of ASPN as a node of cancer progression. And so, but this is a model to summarize our finding in this particular project. It really depends on the environmental cue. If there are canonical Wnt ligand, it turns on the traditional canonical Wnt pathway important for cancer growth. But if the environment are enriched in non-canonical Wnt ligand, it might promote the other way, which is the PCP complex invadopodia and cancer metastasis. So this is quite preliminary but very interesting model we are trying to crack now. So in the last slide, I want to try to share with you a new concept in cancer field. So in the past, if you took your class in cancer biology, you will learn something about oncogene, cancer tumor suppressor gene, right? And they are important for how cancer can grow or form from normal tissue, right? But turns out those genes might not be important for how cancer can progress or kill the patients. There are other subset of genes. They are not important for cancer, how cancer can form, but they are important for how cancer can progress or hide or evade from therapy. Like, again, the most famous case is PD-1, PD-L1, right? If you overexpress PD-L1, it won't give you cancer, right? But it turns out to be a hub or node for cancer immunity. So there is a group of genes. So that is only one example. There are a group of genes. People know more and more about this kind of protein, not important for cancer invasion, not important for normal cell development, but important for cancer to progress or to invade or go to other places. And people give this set of genes a name called cancer fitness genes. You know about fitness, right? 我们叫体适能，这叫癌适能。So we think ASPN is fulfill the definition of cancer stimulus gene, cancer fitness gene. And because it has 3 criteria to be able to call a cancer fitness genes. One is that it has to be important for cancer progression. The other is its overexpression won't cause cancer. And the other is that knockout or knockdown of this gene won't kill the animal. So remember in the very top slide I had shared with you, knockout of ASPN, the mice is viable, right? If you have ASPN mutation, the patients have small brain, right? But they are alive. So meaning that meaning the other advantage of studying this cancer fitness genes that it might be a better cancer target than the oncogene. Because if they are not important for normal cells, if we develop therapy to target this gene, we have a much bigger therapeutic window, right? Or therapeutic index. So that's something that really excited me. Now, let me summarize. So we know that the ASPN is important to control wind, hedgehog notch, and cancer, and also PCP complex for cancer metastasis. That's what we know so far. But there are many outstanding questions there, which is why I'm here. I very happily accept the offer by Director Jen to hear, because I really want to share the story with the expert in this institute to help me out and to help me to move on with this very interesting and maybe important gene. The first question is that why only isoform 1 or variant 1 is upregulated in cancers? Perhaps because remember the only difference is the one single exon, right? So my guess is that there might be controlled by aberrated splicing factor in cancer. Right. Otherwise, if the mutation is shared by both isoforms, both isoforms should go up in cancer, right? But it's not the case. Only isoforms go up. So it must be something other than just simple transcriptional regulation. It must be something important for the splicing event. So that's something beyond the capacity of my lab. And the second question is that why ASPN is the isoform 1 is mainly expressed in the cytosol or cortical region, where the important membrane event is taking place. Maybe there are some vesicular transport are involved. That's my second question. And the third question is that we are not a structural lab, so we want to know. We know about the domain, some domain information about ASPN, the region, the exon 18 region is unstructured based on prediction, based on AlphaFold. So we don't know what to go on from this point on. We need experts to help us. So to figure out why ASPN can interact with so many different binding partners, preventing them from degradation, thereby promoting cancer progression. If we want to go on from here to develop drug or small molecule or whatever approaches, we need to know about how they interact, right? So we need experts to help us to see where we can go from here. And also, the ASPN has been reported as an important regulator in immune cell. So if there is an immunologist here or people interested in immunology, maybe we have a room for further collaborations. And then the last question is that, actually, ASPN is controlling yet another 2 other important development paths, which is ongoing project in my lab. So if anybody of you are interested, you can welcome to come to me. We can collaborate on, because we have limited. We have only 7 person in my lab. I cannot do so many exciting projects all at the same time. So I really want to see, I really can crack this protein in maybe the next 5 years, and I need collaborators to help me out. And I still have another question. And the other very exciting progress that I think maybe it's difficult to solve the structure now, right? But maybe we can take a shortcut approach. We can just take the most very trendy RNA therapeutics, because the technology is there. The carrier, the LNP technology is ready after the COVID-19 vaccine. and there are already 4 siRNA drugs available, get approved. So I put together a team to develop RNA therapy focusing on ASPN right now. So if anybody of you are interested in maybe RNA therapy, please also come to me after the talk, and we can try to see if we can collaborate on that. So finally, I think if we can cut the head of the medusa, we might be able to control tumor all at the same time, right? So you know this guy? Go back. I think his name it's a Greek hero. I forgot the name. He killed the witch in the movie, in the Greek myth. Perseus. His name is Perseus. If you go to the museum, he's everywhere actually in Italy. It's a very famous hero in Greek mythology. So he is the guy who killed the witch. So by cutting her neck, instead of kill the snake one at a time. So I think that might be the strategy to move the cancer treatment field forward from my perspective. So thank you very much, and I think I might took much time, and I have some collaborators in Israel. So Professor Yuval Shekhi is the director in Technion in Israel. He also helped with the project, and also I thank Professor Xu, who used to be my postdoc, and he actually did many of these projects along with my postdoc still in my lab. So thank you very much, and I welcome all your questions and maybe potential collaboration. Thank you.
Okay, so thank you, Dr. Tsai's so comprehensive talk. And so now we learn that in order to kill the medusa, we have to kill it on the right way. We have to kill the snake on her tail instead of we have to cut the half. That is the key point. So I have one question regarding to the ASPN protein. So based on your finding that actually the major or primary those spatial feature associated with this protein ASPN actually come from the exon 18. So for those variants which include the exon 18, they will have all of those oncogenic features as you present here. So that the exon 18 is actually the target.
Right, I think so. So, but when I did, so the review in cancer research, one reviewer asked, I mean, they because right now there's a new class of cancer therapy called a PARP inhibitor, which is important. We're using the strategy called synthetic lithology. And he wanted me to mention more about, because it's the report from the Chinese group, they said the ASPN is stabilized in BRCA1, right? And for so meaning that if you, ASPN might be another strategy to promote the response to PARP inhibitor, because if you inhibit the BRCA1 activity, you might be able to increase the response to BRCA1 to PARP inhibitor. But I replied to him that as far as I know, somehow ASPN interact with BRCA1 in the N-terminal region, not the exon 18 region. But from my research for the WIN, hedgehog, or Notch, the domain, they all reside in the exon 18 encoded region. So that's a mystery to me. So it seems to do similar things to its binding partner, but through different regions. I don't know why. I mean, so that's so I mean, perhaps if we target the isoform 1 alone, we might be able to to inhibit some important functions of ASPN, but not all of them, because some are not dependent on exon 18 encoded region. Well, I mean, I don't know if that answered your question or not.
Yeah, and also that just follow-up question actually is associated with the number one question. So whether those variant 1 is due to the splicing defect.
Right.
And so what is the size of exon 18?
What is the size?
150.
150 amino acid.
150 kilodalton.
But the domain, we know, we have mapped the domain that interact with DVL, right? The Cheval and.
The question will be that because the splicing. So that actually I have to say that. So if splicing
between 17 and 19, so skip 18, but 18 is very long. Oh, if I can go back to the here. So, okay.
So I just would like to
Right.
make it clear is that whether that phenomena is due to the splicing variant, and it's particular for that it is general phenomena or is kind of spatial event, because those variant one or the one which include the exon is only happen in the CSC cancer stem cell.
Oh, yeah, it might be the case because there are some data. We have tons of data. So if you probe the expression level ASP isoform 1, if you compare the cancer stem cell, non-stem cell, it mainly expressing cancer stem cell. So maybe there is some splicing factor that they are controlled by stemness. They are controlling the expression of this splicing event. That I don't know. That's a very interesting question, but it's beyond my ability to, splicing factor is important for cancer, but in Taiwan there are very few labs that can have the ability to, I don't know. I mean, but that's something we can, we can try to, focus on in the future.
Yeah, because there's 2 points of view to address those questions. So number one will be whether those in the CSC cell, their splicing factor somehow is detected, and so they have a global defect for splicing event.
Maybe, maybe.
One the
maybe.
case over here, so there will be one possibility.
Right.
The second one will be whether it's due to the size, because based on the picture the cartoon you draw over there, so it seems like the exon 18 is quite, the size-wise is quite big. So whether there could be another parameter, so maybe the splicing function is somehow impaired.
Oh, right, makes sense.
The impaired function will particularly affect those ones which have long DNA fragment, and so it can cause preferential effect that particular on for these.
Right. Very insightful. Yeah. Thank you. Yeah, I mean there are a lot of potential explanation for this, but those two are very very sound hypothesis, and we can if we have the tool, we can pursue. I mean, anybody I welcome, because I obviously I won't be able to pursue all the questions here, and this is a completely new cancer fitness genes, and I welcome people to jump into the field. I have all the reagent, I have the antibody ready, we have the primer, isoform specific primer, RNAi, everything, CRISPR, right? And so I mean that's my thank you.
Okay. Yeah. So is there any question from our audience?
Yes, thank you.
Thanks, Tsai, for the very great talk. So I have a few questions. The first question is, have you ever examined the DNA sequence of this gene locus? So is there any mutation that will affect the splicing?
You mean if you look at the copy number variation in cancer, right?
Yeah.
And there are very few. We look up the promoter, the sequence, right? There are very few copy number change or promoter sequence change. There are very few. Somehow it's not. I mean, is that your question?
In the gene body, so have you ever checked? Maybe there are some mutation will affect the splicing. So
affect the. You mean the mutation in ASPN affect its splicing itself?
No. For the.
Maybe other splicing factor or.
Mutation affect the splicing, maybe located at the exon 6, 17 or 18.
I'm not sure. I'm not an expert in this field, but my imagination is that maybe there is a aberration, like what Director Chen said. I mean, the mutation or aberration in some splicing factor, but not the ASPN gene itself. But I don't know whether or not the mutation in the protein itself can affect its splicing itself. I don't. Is that a well-known?
Sometimes the mutation on DNA sequence will
affect. Oh, really? Okay. But what we know is that if you look up the mutation profile and the copy number chain, there are very few. There is no, if you look, we can do that easily in using CCGA, right, data, and there are very few, mutation, which can correlate with the expression level I can show you the data later, and of course, I mean, I cannot say in all the cases, but in the majority of cancer, there are very few, mutation in the ASPM gene in cancers, and also no copy number variation or translocation event, very few. So it's something else. It's not, I don't, from my perspective, but I didn't really dig into that, part. Right.
The TCGA did not record the
mutation, right. Mm-hmm.
So I'm curious, maybe.
Yeah, right. We can do, yeah. Welcome to help me. Yeah, right. So, but one upstream we know is the FOXM1. FOXM1, and which is another stem cell factor, and it's also upregulated in many cancers. And if you knock down FOXM1, at least in our case, in gastric cancer, you kill all the ASPM expression. But that doesn't explain why only isoform-1 go up. That's my question. Because it's controlling the promoter, right? But in cancer, only isoform-1 go up. Isoform-2 is still there. So FOXM1 is one regulator, but it doesn't explain everything. I agree with you. So that's why I'm here. So yeah. So there are many interesting questions. I'm not an expert in genetic. So I need some, but I collaborate with a bioinformatic. He replied to me that actually he didn't identify. It's all published actually. There are very few mutations in ASPM gene in cancer, somehow for some reason. So there might be something else, or maybe there are some, mutations that affect its own structure, right? Mm-hmm. And that's I think that's one of the potential explanations for right.
My second question is a very specific question. In your model, the SPM isoform-1 can interact with so many proteins, for the D-several 2, 3,
and also GRI1 and NICD1, NOTCH1.
Yeah, the C-terminal of NOTCH1. So the question is for distractable proteins. So the SPM protein can stabilize it or squeeze it into the plasma membrane. So the next question, for the GRI1 and the C-terminal of the notch, these two proteins must be released and translocate into the nucleus.
Very important question. Right, I don't know the answer to that. Right.
Use different mechanisms.
So. Yeah, right. So I also have the question. We have the same question, right? If it's going to interact with, like, for example, GRI1, which is a TF, right? It has to be translocated and bind to its target gene at some point, right? And how maybe there are other things that regulate how the stabilized GRI1 dissociate from ASPM and move into. I think there are other steps before it really go into nucleus to turn on the gene. But what we know is that when we knock down ASPM isoform-1, the GRI1 level goes to almost 0. But that's not. We didn't do the fractionation. I mean, we didn't really try to to differentiate where that interaction is taking place. Maybe I guess it will be take place mostly in the cytoplasm. But at some point, the GRI1 has to have to translocate into nucleus to turn on the gene expression, right? So at some point, it has to dissociate from ASPM, right? Is that the question? I don't know. I don't know the answer to. The same goes to NOTCH1, right? Or maybe I know. I mean, maybe Professor Chen can tell me. Maybe the protein ubiquitination can also take place in the nucleus, right? Am I right? So I don't know. Maybe it's not all exclusive to the cytoplasm. Yeah, that's interesting. We don't know the answer to that. Yeah, that's a very outstanding question. Indeed, right.
So, any question from students?
Maybe it's a hard topic, very hard, because we also have many. Yeah, sure. Thank you.
I have a general one and not very related to this one. You mentioned this protein is the centrosomal protein in the beginning, right? Centrosomal protein.
Initially, it was identified as a centrosomal protein, but it's only in the M phase. Okay. But in interphase, it's it's mainly expressed in the cytoplasm.
Okay.
Yeah, please go ahead. Sorry.
Because this protein is related to like brain size growth and tumor size growth. So how does this protein related to like cell cycle or cell division, or is it not related?
That's I'm not sure if I capture your. So I think what we know is that the isoform 2 are actually the protein. Those NCB paper they are studying, because if you look at the size of the publication, the size is small, like 250, not the isoform 1. So we guess it's the isoform 2, which is, we actually have a paper on that, right? It's the housekeeping isoform 2 is regulating cell mitosis. Only isoform 1 is regulating development and stemness pathways. So that's our understanding at this point. But I don't know. I mean, during interphase, I mean, the protein, some of the protein, maybe isoform 1, they are expressed in the cytoplasm of neuroprogenitor cell, which is stem cell-like, right? And so they are stabilizing cyclin E to allowing the cell cycle to progress from G1 to S checkpoint, right? R point. But we don't know whether that's also important for like differentiated cells or normal cells, because that paper is studying neuroprogenitor cells. And I don't know how important that mechanism is for normal cell, for differentiated cells. I guess it's not as important as in the stem cell or progenitor cell. I mean, but there are other, I think we have a project going on. We are trying to compare how important ASP is for the cell cycle in stem cell and not stem cell. So that's one project. That's also my curiosity. Is that your, yeah. So any student interested in cancer, welcome to, to talk to me. And so for the oncologists like me, what we really want is not to publish the paper. We want to develop something to help patients, right? And the cancer field, so far, beyond the so-called checkpoint immunology, nothing big happened in this field for almost like in recent 5 years. No, I mean, maybe there are some, precision medicine drug, but it only helped a minority of patients. We need something, the next big breakthrough to help the cancer patients. So if you are really interested in the field, I mean, welcome to talk to me. And I think there's still a lot of questions to be answered in the field of cancer. Thank you.
So I think quite important, or what we have learned today is that actually in the past we always want to look at for one particular gene and to try to find out how to able to manipulate a particular one gene. But turn out that actually the cancer cell is so smart, so we need to find the key. And the key is not single gene, but it's the hub. Would be that the hub is quite important in the field, because if we would like to treat the patient, we need to find the hub. And also we would like to learn what is the mechanism associated with the action of the hub over there. And I think that is very important for the future, near future of the cancer treatment. And we are very grateful that Dr. Tsai have both shared all his findings in the past decade and all his important insight. So let's give a big hand to Dr. Tsai again.
Thank you. 谢谢。要照个相先。